So today we'll be discussing about a very important and a very relatively common flow cytometry assay, which is diagnosis of PNH, paroxysmal nocturnal hemoglobinuria, by flow cytometry. Right? A very important assay and a very simple assay also. And let's see how we diagnose PNH by flow cytometry. So, you know what is PNH? PNH is a rare hematopoietic stem cell disorder. Okay, it is an acquired hematopoietic stem cell disorder. I mean, like, so the problem here is there is a mutation in the PIGA gene, which is located in the X chromosome. Okay, now when this gene gets mutated, in wild or in normal conditions, this particular PIGA gene will encode for a anchor protein called glycophosphatidylinositol. Okay, that is called GPI. Okay, so in normal conditions, this PIGA gene located in the X chromosome will encode for a protein called GPI protein. Okay, the function of this GPI protein is that this GPI acts as an anchor to hold on some of the cell surface proteins to the cell membrane. Okay, so if you see cell surface proteins, some proteins are transmembrane proteins. That is, the proteins will be present throughout the span of the cell membrane. That is, it has a cytosolic part, it has a membrane part, and it also has an extra membrane part. Okay, whereas some proteins, surface proteins, will be linked to the cell membrane by some anchors called GPI. Okay, so that is called GPI anchor. And this GPI anchor, as I said you, this GPI protein is encoded by a gene called PIGA gene, which is located in the X chromosome. When there is a mutation in this PIGA gene, now this leads to complete or partial loss of this GPI anchor. So when there is loss of this GPI anchor, obviously the proteins which are linked to this particular GPI anchor will not be present. Okay, that leads to certain cell surface protein deficiency. Okay, these proteins have a protective function for the cell, and when these proteins are not there, they may be lead to cell damage or even cell death. Okay, so that is the basic pathophysiology of PNH. Okay, so now as we discussed, like as we know, the PNH typically presents with three clinical features or a triad of features. One is the patients present with intravascular hemolysis, which is basically because of complement mediated. Okay, so as I said, this GPI link proteins, some like CD55 and CD59 in RBCs, have a protective role to save the RBCs from complement mediated lysis. When this GPI protein is not there, now this protein CD55 and 59 will not be there on the cell surface. When such a RBC is very susceptible to complement mediated lysis, and they undergo lysis when they are in circulation. So that is the first classic feature of PNH is predominant intravascular hemolysis. Second is a characteristic thrombotic tendency. Okay, and third, since it's a stem cell disorder, okay, it's a hematopoietic stem cell disorder, this may also leads to bone marrow failure. So this classic triad is very classic for PNH or the diagnosis of PNH. Okay, now this is what I said you. So some cell surface proteins will be spanning throughout the membrane. So you have and such proteins are called transmembrane proteins. Okay, so if you see this protein, it has an extramembranous part, a membranous part, and a cytosolic. Whereas, as I said, some proteins where it will be linked to the cell membrane or cell membrane by an anchor protein called GPI anchor. Okay, now this is the protein. Now this protein is not directly linked to the cell membrane as the transmembrane protein. Now this protein is linked to the cell membrane by an anchor protein called GPI anchor. So the GPI anchor will hold the protein to the cell membrane. Okay, now in certain conditions, this particular protein has a very important function in saving and protecting the cell. Okay, when it is lost, the cell may undergo damage or even cell death. Okay. Now, this is normal condition. In PNH, as I said, there may be a mutation in the PIGA gene, and this particular protein will not be encoded. So when this protein is not encoded, obviously this particular protein will not be sticking to the cell surface or the cell membrane, or it will not be attached to the cell membrane. So there is loss of these proteins, which may lead to cell damage or cell death. Okay, so this is the pathogenesis of PNH. Okay, so another representation. So the PIGA gene encodes for GPA anchor. This is the GPA anchor, and this GPA anchor is essential for the attachment of some proteins like CD59 and 55 in RBCs to the cell surface. Okay. Now, if there is a mutation in the PIGA gene, now this GPA anchor will not be there. So, since the GPA anchor proteins are not there, there will be no such cell surface proteins to, in case if there is no GPA anchor, there will be loss of these cell surface proteins, and these proteins will not be expressed on the cell surface. So now, how do this help in flow cytometry? Now, the defining phenotypic feature of PNH cells is the deficiency of proteins which are linked by the GPA anchor to the cell surface, cell membrane. Okay. So we will be exploiting this feature for the diagnosis of PNH by flow cytometry. So how do we do? We have already seen that the beauty of flow cytometry is that we can characterize any protein or any antigen if we have a complementary antibody tagged to a fluorochrome, right? So the basic idea of diagnosis of PNH by flow cytometry is we'll be targeting these proteins. Okay, so we'll be using an antibody tagged with fluorochrome against these particular GPA linked proteins. In this case, it is CD59 and CD55. So I'll be using an anti-CD59 tagged to fluorochrome, and I'll use anti-CD55 tagged to a different fluorochrome. When I add, these antibodies will come and bind if the cell is having GPA proteins, that is in normal state. If the patient is a PNH patient and there is no GPA anchor, these proteins will not be present on the cell. So these antibodies will not bind to PNH clone or PNH positive cells. Okay. So when we run it in the flow cytometer, when we get the signal for this protein, it means that particular protein is present and that particular cell is normal. If we are not getting the signal, then that particular cell does not have the protein, the GPA linked protein, and that is evidence for that it's a PNH cell or a PNH clone. Okay, so this is how we are going to use flow cytometry in the diagnosis of PNH. Okay, so again some more illustrations. So this is normal RBCs and this is the GPA anchor, and there are two important proteins which are linked to the RBCs by this GPA anchor. One is CD55, which is called decay accelerating factor. The other is CD59, which is MARL, membrane inhibitor of reactive lysis. So these two important proteins are linked to the RBCs by the GPA anchors. Okay, in case the patient is a PNH patient and there is a mutation in the PIGA gene, now these GPA anchors are not produced, and since the GPA anchor is not there, these particular proteins will not be bound to the cell and it will not be there on the cell surface. Okay, so when such a cell is very susceptible for complement mediated lysis. Okay, so this is again a similar representation. So we have CD55, which inhibits C3 convertase and which inhibits C3 convertase, and we have CD59, which will inhibit the membrane attack complex, and that's how saving the RBC from complement mediated lysis. Okay, now so what I said I'm just going to show you in a pictorial representation. So this is the cell, and if it's a normal cell, it has a GPA linked protein. Now what we do, we use an antibody against this particular GPA protein, which is tagged to a fluorochrome. So once we add it, this goes and binds to the cell, and when we run it through a flow cytometer, we get the signal. So when we get the signal, it means that it's a normal cell and the GPA linked proteins are present. And when we are not getting the signal, that means the protein, the GPA linked proteins are absent, and we diagnose the PNH flow by that. Okay. So flow cytometry is the method of choice to detect the loss of GPA linked proteins. Okay. Any doubts till now? Any doubts? So far it is clear?
Sir, if there is no signal, then it is PNH.
If there is no signal, yes, then it is PNH. It is a PNH flow.
Okay, sir.
Correct now? So see.
So what is CD55 and 59? What did you tell? The names?
So it's decay accelerating factor. See, I am just telling out from my memory, like I would have read during my post-graduation. So it's decay accelerating factor, CD55, and CD59 is membrane inhibitor of reactive lysis.
Okay, sir. Thank you.
My theory, I mean, like it's been long time. Okay, fine, right? So as simple as that. If there is signal, then the GPA protein is present. So the cell is normal. If there is no signal, then that particular protein is not there, which indicates that that's a PNH flow or a PNH cell. Right. So these are a list of, I told you what is CD. Whenever there is a prefix CD with a number, it denotes a, anyone?
So cytoplasmic protein.
No, you just said ulta.
Okay, surface.
Cluster of differentiation.
So only surface markers will be designated as CD. Okay. Only surface markers will be designated as CD. You don't find any cytoplasmic protein or antigen designated with the name of CD. Okay. You may say, sir, how about cyto CD3? Cyto CD3 is again a surface. It was characterized on the surface, but during its immature stage, it may not be put on the surface. It will be present in the cytoplasm. That's why we call cytoplasmic CD3. But still, it has been first characterized on the cell surface only. Okay. So, for example, TDT, MPO, we don't have a CD name for that. Okay. So, whenever you have something, a marker with CD, it means it's a surface protein. It's a surface protein. Okay. Now, these are some of the surface proteins or CD molecules which are GPI linked. Which are GPI linked. So, in PNH patients, the cells will not have these particular antigens or proteins on that cell surface. Okay. It depends upon the cell type also. Okay. For example, if a patient of normal monocytes or a patient, a normal monocyte means monocytes of a normal individuals do express CD40. Okay. In patients of PNH, the monocyte of PNH patients do not express CD14 or express CD14 in a lower level. Okay. So these are some of the markers or antigens which are GPI linked. Okay. So we may not be exploring all the GPA linked proteins for flow cytometric diagnosis. We'll be selecting only specific ones. So for monocytes, we'll be using CD14. Okay. For neutrophils, we'll be using CD24. For RBCs, we use CD55 and 59. Okay. So you can ask me, sir, why we are not using CD16 for neutrophils, though CD16 is a GPA linked marker. And similarly, you can also ask lymphocytes, where CD52 is a GPA linked marker. But these markers are more robust. Okay, and it is easy to demonstrate their presence and absence by flow cytometry when compared to the other markers. Right? So I repeat, for monocytes, we use CD14. Okay, so monocytes of a PNH patient may show loss of CD14 in monocytes or decreased CD14 expression in monocytes. Similarly, neutrophils, we use CD24, which is GPA linked. So in normal conditions, CD24 is positive in neutrophils. In neutrophils of a PNH patient, CD24 will be lost or downregulated. Okay. Similarly, for erythrocytes, we use CD55 and 59. Okay. So, but I insist upon again that there are multiple other PNH linked or GPA linked proteins which we are not using in flow cytometry, but there are multiple GPA linked proteins which I have some of them which I have shared. Which I have shown you in this particular list. Okay. So recently, one more marker was introduced, like some 3 years back, which is CD157. Now, the beauty of CD157 is that, see, if you want to study monocytes in PNH patient, okay, you have to use CD14. And similarly, if you want to study neutrophils in a PNH patient, you need to use CD24. You cannot use CD14 for neutrophils or CD24 for monocytes. You understand? So CD14 is only for monocytes and CD24 only for neutrophils. Okay, because CD14 is expressed in monocytes and not in neutrophils, and CD24 is expressed in neutrophils and not in monocytes. Okay, so you need 2 markers, and each marker is expressed in only either monocytes or the neutrophils. Now the recently introduced CD157, the advantage of this particular GPA linked protein is that it is expressed in both neutrophils as well as monocytes. So when you have CD157 in your panel, you can study the GPA status in both PNH status in both neutrophils and monocytes. Understood? That is the advantage of CD157, which is also being used in flow cytometry for the diagnosis of PNH. Okay. Now, apart from CD14 in monocytes, CD24 in neutrophils, and CD157 in neutrophils and monocytes, a very important marker for the diagnosis of PNH by flow cytometry is FLAIR. Okay, FLAER. So the full form is fluorescent label pro-aerolysin. Okay, fluorescent label pro-aerolysin. Okay, now what is this FLAIR? Okay, now let me tell you before what is FLAIR. Pro-aerolysin is a prototoxin or an inactivated form of toxin aerolysin, which is a virulence factor of bacterium Aeromonas hydrophila. Okay, so now what happens is that, so when the pro-aerolysin, after proteolytic nicking at the C-terminus by an enzyme present on the cell surface, okay, gets activated to form aerolysin, which is the virulence or an active form. Now, this active form generated binds to GPA-linked cell surface structures, oligomerases, create channels and result in cell lysis. Okay, so for the aerolysin to kill the cell, it needs to bind to GPA-linked cell surface structures. So this is the actual pro-aerolysin or aerolysin and how it attacks the cell. So we understood this, and what we later did, since it is using this GPA-linked cell surface structures, this particular aerolysin, we developed in vitro for diagnostic purpose a modified form of pro-aerolysin. Okay, so as discussed here, once it gets activated by the proteolytic nicking, it turns to aerolysin. But now this modified aerolysin cannot kill the cell. Okay, it cannot kill the cell, and we use this particular thing for diagnosis of PNH. So subsequently, a fluorochrome conjugated version of non-lysing mutated form of pro-aerolysin was generated. That is called FLAIR. So what is FLAIR? FLAIR is a fluorochrome conjugated version of non-lysing modified or a mutated form of pro-aerolysin. Okay, that was generated, which retained its specificity for GPA-linked structures without causing cell lysis. Okay, and we are using this as a very, very important marker for the diagnosis of PNH. Okay, is that clear? Any doubts? Any doubts? So FLAIR, you are understanding, right? So FLAIR is a very important marker in PNH. So any lab which does diagnosis of PNH by flow cytometry will use flare. Will definitely use flare. The beauty of flare is that I told you like CD14 only for monocytes and CD24 for only neutrophils. CD57 you can use for monocytes as well as neutrophils. See, you can use flare similarly in neutrophils, monocytes, even in lymphocytes. Any WBCs you can use this. So that is the beauty of flare. Okay, so I repeat, what is flare? Flare is a mutated or a modified form of pro-aerolysine which is fluorochrome conjugated. Okay, the fluorochrome which is commonly used is Aloxafluor 488. Okay, so it's a modified or a mutated form of pro-aerolysine which has been fluorochrome conjugated. So we use it for diagnostic purpose. So when you add this reagent, it goes, okay, it gets activated and it binds to GPI linked structures. If the patient is a PNH patient, then it may not be able to bind to the cell surface because there is no GPI linked markers and there will be no signal with flare. If it is normal, you get signal with flare. Okay. So fluorochrome conjugated version of non-lysine mutated form of pro-aerolysine is called flare. Okay, that has specificity for GPA linked structures without causing cell lysis. The wild form or the non-mutated form will kill the cell, but we modified in such a way that we can use it for diagnosis. Okay. Now flare, as I said you can use in any WBCs, but you cannot use in erythrocytes. The reason is for flare to work, the pro-aerolysine should be converted to aerolysine. And for this conversion, you need an enzyme that causes the proteolytic nicking at the C terminus. That particular enzyme is not there in RBCs. Okay, so this surface-bound proteolytic enzyme which is present in WBC is not present in RBCs. So you cannot use flare for the diagnosis of PNH in RBCs. Okay, you can use in WBCs, but you cannot use in RBCs because RBCs do not possess that enzyme which is needed for the conversion of pro-aerolysine to aerolysine. Okay, and one more point is, you know glycophorin, which is a glycoprotein present in RBCs. Okay, it binds weakly to pro-aerolysine, non-specifically binds to pro-aerolysine. So it will give you a false positive signal. Even a PNH patient can give you a false positive signal for flare when you're using in RBCs. So that is the reason why flare is not used in RBCs and used only in WBCs. So you can use in any WBCs. It's not like only a CD14 only in monocytes and CD24 only in neutrophils and 157 where monocytes and neutrophils. Flare can be used in any WBCs. But of course, for diagnostic, we'll be testing for PNH clone only in neutrophils and monocytes, and of course RBCs. So three populations for which we check for PNH clone is, note down, neutrophils, monocytes and RBCs. Okay, good. Now, so these are some of the GPA linked antibodies or proteins that are commonly targeted in clinical flow cytometry. For neutrophils, we'll be targeting CD24, which is present only, which will be used only in the neutrophils. Then you have 157 and flare. Similarly, for monocytes, we have CD14, which is used only in monocytes. We have 157, which we can use it for both monocytes and neutrophils. And similarly, flare, which we can use in neutrophils and monocytes. Okay, for RBCs, only 55 and 59. You cannot use any of these markers for RBCs because flare is not helpful in RBCs for the simple reason what we saw in the earlier slide, and these markers are not expressed in RBCs. So we'll be using only 55 and 59. Okay. Now this is a normal blood or a normal control. So here I have shown you the RBC population. Here I have shown you the neutrophil population, and here I have shown you the monocyte population. So if it is a normal RBC, it will be expressing CD55 and 59 in a normal fashion, and you don't find any PNH clone which is negative for CD59. See, this area is vacant, empty. Similarly, for neutrophils, you see this is normal neutrophils which express the GPA linked protein CD24, and it is also positive for flare. And you don't find any neutrophils which are negative for 24 and flare. Similarly, this is monocytes, which is expressing CD14, which is a GPA linked marker for monocytes, and it's also expressing flare, signal for flare, and you find no population in the double negative area, which is indicative of PNH clone, but here it is absent. So there is no PNH clone, only normal monocytes are there. In contrast, this is a patient sample, a PNH patient sample, where you find that a subset or a small population of RBCs, though you find a normal RBCs, a small population of RBCs are lacking a PNH GPA linked protein CD59, okay, amounting to 1.3%. Okay, so there's a small clone which is a small PNH clone is present, which is showing absence of CD59. Similarly, neutrophils, though the predominant population is normal, expressing 24 and flare, you find a small subset of population of neutrophils which are negative for 24 and flare. Okay, similarly in monocytes, you find a small subset of monocytes which represents the PNH clone showing negative for 14 and flare. Okay, and these are normal monocytes. So that's how we diagnose. Okay, so what are the technical considerations in PNH? So the anticoagulant, what we commonly use is, as we use for other assays, we use either EDTA or heparin. So the preferred sample volume is minimum of 1 ml or 3 ml. Adequate or 3 ml is more adequate, but some cases where the patient is as set in an aplastic anemia or an hypocellular MDS. So even PNH testing is done in these cases, and when the patient is having pancytopenia, because of this, you need larger volume of blood because it's hypocellular sample, right? So when you lyse and you need at least some WBC, so you need larger volume of blood. If the cell count is good, then 2 ml is sufficient for the assay. What is the maximum sample age? For WBCs, as with any leukocyte assay or any clinical flow cytometry assay, for WBCs, it is less than 48 hours is desirable. But for RBCs, a 7-day sample, less than 7 days is optimal. So even in the 4th or 5th day after taking the sample, you can do PNH assay on RBCs, but not on WBCs. So the sample is stored in 4 degrees for 24 hours. So lysing reagent, so you know the 3 ways of processing a sample in flow cytometry, or the 2 common ways. One is stain lyse wash and other is lyse stain wash, right? Here, if you are doing in WBCs, you can use stain lyse wash protocol. But if you are doing PNH assay on RBCs, you should not lyse the RBC. You should skip the lysis step. Okay, since the population of interest is RBC, you should not be lysing the RBCs here. So you should skip the lysis step. Okay. So sensitivity of the routine assay is 1%. So if you want to detect PNH clone greater than only greater than 1%, then it is sufficient for you to acquire somewhere around 5,000 events. But most of them it's better you do an high sensitivity analysis where you will detect PNH clone beyond 0.01%. So if it's the case, then you need to acquire more number of events. So around like 250,000 events has to be acquired. Okay. So some minor clones will be missed when you are acquiring smaller number of events, like 5,000, 10,000 or 50,000. So when you acquire 250,000 or 5 lakh cells or 1 million cells, then you can pick up much lower percentage of clones or a smaller clone, PNH clones present in the patient, which is really good. Okay. So the cell populations used in routine analysis, as I said, is granulocytes and monocytes. Okay, so you should definitely see it in granulocytes and monocytes. If granulocytes and monocytes shows a PNH clone, then you need to check the PNH clone on RBCs. I repeat, when you are doing the PNH assay, when you detect a PNH clone in the neutrophils and monocytes, that is, you are finding a monocyte population which is negative for 14 and flare or 157. And similarly, you're finding a population on neutrophils which is negative for 24, flare or 157. Then you need to do pNH assay on RBCs. Okay, so you need to check whether the RBCs are also showing pNH clone. If the neutrophils and monocytes does not show a pNH clone and appears normal, then RBC assay is not needed. So that is what the recommendation says. Is that clear? Okay, so these are some of the technical considerations you should bear in mind when you are doing a pNH assay. Okay, so is that clear? Any doubts? I'll just 30 seconds. Any doubts? You can ask your doubts.
Sir,
Yes.
minimum how many percentage of pNH clones should be there to call it as positive?
I'll discuss that in the last slide. Okay, I have that for you.
Okay,
It is a part of the session. I'll discuss later. Okay. So the point what I want to say with that slide is, if you want to detect 1%, it depends upon your sensitivity of the assay. If you want to detect only beyond 1%, it is enough if you acquire 5,000 or 10,000 WBCs. If you want to detect a minor pNH clone, which is somewhere from 0.01% to 1%, even if you want to cover that particular range, then you need to acquire more number of events. Say, for example, 5 lakh events or 1 million events. Any other doubts? So is that clear now? So no doubts. Can I go ahead? Right. Thank you. If you have any doubts, please put it in the chat box. Now, let's go through how we do pNH analysis on RBCs. Okay. Now, so whenever you're doing an analysis on either RBCs or neutrophils or monocytes, you need to use a gating marker. What is a gating marker, anyone? We have finished panel designing, so someone has to tell me what is gating marker.
So like selecting the population or areas of interest, like we are doing like CD45 versus site scatter. So then we are selecting the area of
Yes.
like CD45
Yes.
negative region, and then we are putting it in the next plot.
Perfect. So gating marker is a marker which will help you to isolate a population of interest for further analysis. Okay. Now, for example, someone tell me a gating marker for monocytes. You can give blind guess. Like monocytes, what is a common marker used?
64 or CD14?
64 or CD14. Okay. Now, see, CD14 is very good. It's a gating marker, but you should not use it as a gating marker in pNH assay. Why? Because CD14 is a GPI linked marker. If you are using that to get the monocytes, okay, if it's a pNH patient, then the monocytes will not express CD14 and you will miss the population. You will not take the monocytes for further analysis. Okay, so that is what. So you should use a gating marker for pNH assay to segregate or isolate the neutrophil monocytes or RBCs, but make sure that the gating marker is not a GPI linked marker. Understood? So CD14 you should not use for monocyte gating in pNH assay. You can use it for some other assay. Some other monocyte assay you can use. But in case of pNH assay, you should not use 14 for gating the monocytes. Understood? Similarly, you should not use 24 for gating or separating the monocyte. Neutrophils. 24 you should not use for separating the neutrophils. Okay, that point is important. So you need a gating marker to isolate the population to take it for further analysis. Okay, now in routine analysis, RBCs can be identified by the light scatter property. So normally RBCs, platelets, we see it in forward scatter, forward scatter and side scatter in logarithmic scale. Okay, normally I told you scatter properties we study for WBCs in linear scale, but for platelet RBCs we use logarithmic scale. Okay, now once you find the RBC population, because we are using logarithmic scale, because the separation between the platelets, RBCs and debris will be quite good. Okay, so once you find the RBCs in the with the scatter properties, you need to refine that RBC and you need a gating marker to take only the RBCs and eliminate all other non-specific population. So we use the gating marker CD235A, otherwise called as glycophorin. So after selecting the RBCs from the scatter properties or from forward scatter, side scatter, I put that population in side scatter CD235A and I'll take up only the population expressing CD235A for further analysis. I will eliminate all the population which is negative for CD235A. Okay, so that is the gating marker here. Okay, now and I'll show you. See, this is how. So this is, time for stable acquisition, and then in forward scatter, side scatter, you can see the cluster of RBCs. Okay, once you get this particular cluster, you check for the expression in 235A. You need to select only the population which is expressing 235A for further analysis. You should eliminate all these population which is not expressing 235A, because that could be some junk which is coming inside this. Okay, so that is the use of a gating marker. Now once the gating marker is done, you study this population for the expression of CD55 or CD59. Okay, so when you study that, you find three types of RBCs. One is type 1 RBCs, which are nothing but normal RBCs, which shows bright expression of CD55 and CD59. Okay, so those RBCs are called as type 1 RBCs. Second, you find, if it's a PNH patient, you find RBCs which show complete loss of CD59 and CD55, or complete deficiency of CD59 and 55. Okay, so these RBCs have no protection from complement and they die within 10 to 15 days. Such RBCs with no expression of CD59 and 55 is called type 3 RBCs. Okay, now you know what is type 2. Type 2 is RBCs which shows some expression of CD59 and CD55, but not as to the level of normal expression. Okay, so type 2 PNH RBCs have partial expression of CD59 and 55 deficiency. So there will be some expression of expression of CD55 and 59. Their lifespan is intermediate between type 1 and type 3 PNH RBCs and depends on the level of CD59 deficiency, often as a result of multiple mutations. Okay, so these type 2 RBCs have some partial protection from complement. So you know how to get the RBCs now, and then how to segregate the RBCs as type 1, type 2 and type 3. So if you take a normal control and run the PNH assay for RBCs, the population what you find is only type 1. Okay, so in PNH patients you may find type 3 or type 2. Okay, even if some normal RBCs are all, if it's a partial thing, you can also find some type 1 RBCs in PNH patient. Okay, now this is a PNH patient where I'm showing you an histogram of RBCs showing CD59 expression. Now you find, as I showed you, there are three populations. One population is showing normal or bright CD59 positivity in this particular peak, which is around 16%, 16.8% of total RBCs. Okay, 16% of RBCs are showing normal expression of CD59. Okay, these are type 1 RBCs, which is seen in normal patients. Which is seen in normal individuals or controls. And then you find a population, in this case 11.9% of RBCs, where it is completely lacking CD59. Completely lacking CD59, or it is complete deficient for CD59. This is type 3 RBCs. And then you find another population, in this case it is 71%, where there is some expression of CD59, but not to the level of that is seen in normal condition. So these RBCs have some partial protection against complement because they show some expression of 59, and these RBCs are called as type 2 RBCs. Okay. If you ask me, you select one among the fluorochromes. Someone will ask a question like, which is better, 55 or 59? 59 is better than 55. The reason is CD55 is less abundantly expressed on red cells and not recommended as a sole reagent because it frequently does not provide adequate separation between the type 1, type 2 and type 3. So 59 is always better. If you are using only one marker, better use CD59. If you are using two, then you can use 59 and 55. You should not use 55 alone because that separation may not be that great with 55. Okay, so I show you this. Like first you ensure that there is a stable acquisition with time plot. Then you gate the RBCs with forward scatter, side scatter in logarithmic scale. Once you have gated this, purify this particular population so that with a gating marker which is CD235A or glycophorin. So you select only the RBCs, eliminating all other non-RBCs which has come from this gate. Once you have purified, you gate this population on for the expression of CD59. So here you find that the entire population is expressing CD59 and there is no population which is showing loss or diminished expression of CD59. So what I mean to say, there is no type 3 or type 2 population. You are finding only type 1 population. So this is normal. Okay. So this is normal. Now this is a sample of a PNH patient. Similarly, after ensuring a stable acquisition with time plot, I have gated only the RBC. I have gated the RBCs using forward scatter and side scatter plot. And then I'm refining the gated RBCs with the gating marker, which is 235A. And after gating only the RBCs, now I'm gating it on CD59 with 235A. Now, if you see, this particular RBCs are splitting into three populations. One population is type 1, where it is expressing CD59 in normal amount. Then you find another population of RBCs which is showing complete loss or complete deficiency of 59, which is type 3 RBC population. And then you find a small population between these two, which is type 2, where it is expressing some amount of CD59. Okay, so and when you are reporting, you need to report all the three populations. Okay, you need to report as PNH clone detected in RBCs by adding these two. And you also need to report separately type 2, type 3. Okay. Now, this is again a case. Similarly, I'm showing you. So, this is time gate with forward scatter, side scatter. Just gate the RBCs. Then purify it with a gating marker like 235A. And then gate it across 59. Again, you're finding three populations. One cluster. This is scatter plot. This is a density plot. Okay. You find one cluster is normal, expressing normal levels of CD59, which is type 1. Then you find another cluster which is showing complete loss of 59, which is type 3, and then an interim population which is showing some level of CD59 expression, which is type 2. Okay, so when you are reporting this case, you should report like PNH clone detected in RBCs, which is equal to add type 3 and type 2 together. So in this case, it may be around 60%. Okay, and you also need to report that type 2 is 22% and type 3 is 37%. Okay, because patients with type 3 will show symptoms and you need to immediately treat them. Okay, whereas type 2, these patients may not show symptoms and may do well. Okay, that is the reason why you need to report these two clones separately. Okay, now this is again, I'm showing you another way. I'm showing now in an histogram. So you find, so once after gating the RBCs with an RBC gating marker 235A, I have put the population in CD59. You find this is normal expression of 59. So this is type 1 RBCs. Then you find a type 3 RBC showing complete loss of 59 and a type 2 which is showing some expression of CD59. This is control normal. Then you need to give the percentages. This is histogram where this is type 1, this is type 3 and this is small clone is type 2. this may appear very easy for you, but how do you position the quadrants? Now, see, from here to here is type 3, from here to here is type 2, from here to here it's type 1. Since you're getting the population as a cluster here, it is easy to place. But during your routine analysis, it may not be very easy. So, how do you place the quadrants? Anyone? Anyone wants to give a try? How do you place the quadrants? How do you place that? From here it is type 1. From here till here it's type 3. From here to here it's type 2. So how do you place the quadrants? Any blind guesses? Yes, Dr. Bishal.
So there is a divider option there. With that, can we use?
Yeah, of course, the software you have a divider you can use, but how do you set it up? This is my question. So what you need to do when you are doing, you run the normal control. Control is normal individual. So there, the normal individual will show only type 1, correct? It will show only type 1. So you draw the quadrant for that as type 1. Then what you do, you run the same control sample without staining it for CD59 or 55. So when you have not used that antibody, what happens? The antibody you have not used, unstained. So you get a big peak which will represent something like type 3. Because there is no antibody you have not added. You have intentionally not stained the cells and you are acquired it in flow cytometer. Now you get a big peak here. Okay, because of no antibody. Okay, so that you get it as type 3. So you got type 1 and type 3. Then in between you can draw a divider and make it as type 2. That's how you position the quadrants. So when you are acquiring a patient sample, you acquire in that particular template so that you can able to check whether it is type 1, type 3 or type 2. Clear?
So please repeat it.
Yeah. So what you do is that, so my question to you is that, how do you position these quadrants? For example, now I have beautifully drawn a gate here saying like whatever falls here is type 3. And similarly here, whatever falls here is type 2 and whatever falls here is type 1. Now, positioning this may not be as easy as I've shown here. So to do this, you need to do a small experiment like thing. What you do, you take a normal individual or a control sample, run it. Okay. The control will obviously show you only type 1 cells because they show normal expression of CD59. So you will get only this peak and you get only this population. So you draw this one and label that as type 1 cells. Then you take the same sample. Now you don't add any antibody like CD55 or 59. Since you have not stained the cells, the entire population now will come in this area. Correct? Now you draw a gate for that and label it as type 3 cells. And in between this type 3 and the type 1, you draw a gate and label it as type 2. That's how you can able to set the quadrant. Now if you run the patient sample in the same protocol or the template, if the population is lying here, it is type 1. If it is lying in this area, it is type 2. And if it is falling here, it is type 3. That's how you can able to segregate the RBCs. Clear? Is that clear?
Yes, sir.
Perfect. So that's how. So positioning the quadrants and regions. A negative control consisting of unstained cells is often useful for identifying the expected position of type 3 cells because you are not adding any antibodies. And it behaves something like RBCs of a PNH patient. If a PNH sample is mixed with approximately stained normal sample, and this is some way we can do. The one way of doing is, as I said, run a normal stained control and an unstained control. Those two will represent you a type 1 gate and a type 3 gate respectively. The other way is, when you're running a PNH sample, you can mix it with some stained normal sample. Now, since you have already you have spiked it up with some normal sample, you will find a type 3 population sitting. So with that, relatively, you can find how is the expression in PNH patient. Okay. So the expected position of the type 2 cells can be identified as the area between the positive and the negative population. Okay. Histograms are preferred for quantification of type 2 and type 3 cells. So when you're quantifying, it is better you quantify them on histograms and not on dot plots, as we do here. So always you quantify on histograms. Okay, so when you get a pure PNH patient where there is no expression of 55 or 59, you spike it up with some normal samples. So you get two clusters. One will be the type 1 of the normal RBCs, and you'll get type 3 of the PNH RBCs, and in between can act as a type 2. Once you have made this gate ready, you can use this template or protocol for acquiring the patient samples next time or the subsequent times. Okay. Now, there are some technical things which I want to tell you. Washing the RBCs more than once can help to decrease non-specific binding. Okay, now see, this is somewhere you find how many peaks are there here. So, how many peaks do you see? So, as I said you, by doing this experiment of running normal controls and unstained samples, I've drawn the gates like type 3, type 2 and type 1. Now I'm running a patient sample. So how many peaks are you seeing?
2.
Very good. Okay, but the separation is not that great, right? So when you wash the RBCs multiple times, then the separation will be much better. Now you find that the population is type 3 is 53.2%, whereas here it is negative. So doing RBC, when you're doing PNH assay on RBCs, is technically more demanding than doing a PNH assay on WBCs. I repeat, doing a PNH assay on RBCs is technically more demanding than doing a PNH assay on WBCs, which is easy, relatively easy. Okay, so the problem, one of the problem is this: if you're not washing it properly, there will be some non-specific binding and you get a false positive 59 positivity. But once you wash it properly, then you find that the huge type 3 cells are there and type 1 cells are there. Here it appeared like there is some type 2 cells and type 1 cells, which is wrong. Similarly, RBCs tends to get aggregated. Okay, when you add antibodies, glycophorin and CD59 or 55, these RBCs tends to aggregate. So when you wash it, the aggregates may decrease. Okay, the aggregation may get decreased. Similarly, you vortex it. Vigorous vortexing or running the tube across a test tube rack just to give some vibration can disaggregate the cells. Okay, you know what is the dictamen flow cytometry, the first class. To have a good flow cytometry assay, one cell should pass through the laser beam at a given point of time. When the RBCs aggregate, what happens? It will be like a 3 or 4 clumping of RBCs going at one particular time, which will give you a very bad signal. Okay, so when you vortex it or running the sample across a test tube rack, it helps in disaggregating the RBCs. Okay, so red cell agglutination decreases substantially when the red cell antibodies are diluted from the manufacturer recommended concentration. So when the manufacturer said 20 microliter, don't add 20 microliter. You titrate it and use it in a diluted form so that there will be not much of RBC aggregation. Okay, so that exercise you need to do. Okay, then reporting of PNH RBC clones, report the clone size in red blood cells, including both total PNH clone size as well as the percentage of type 2 and type 3. This I told you, right? So you need to report the total of type 2 and type 3 and also report separately type 2 percentage and type 3 percentage. Okay, because there's a clinical significance associated with the proportion of type 2 cells and type 3 RBCs. Okay, since the clinical significance of type 2 versus type 3 PNH RBC is well established, you need to report the total clone, which is the sum of type 2 and type 3 RBCs, PNH RBCs, and you also need to report separately type 3 RBCs, which is complete loss of PNH proteins or which is CD55 and 59, and you also need to report type 2 where there is partial expression of CD55 and 50. Okay, so this is why it is said so, because when there is no specific cutoff that determines when a PNH patient is likely to show symptoms, but it has been published like when the type 3 cells are greater than 20%, they likely show clinical signs and symptoms associated with intravascular hemolysis. Okay, in contrast, patients with large type 2 populations, in the absence of significant type 3 populations, may show a reticulocytosis, elevated LDH, but less hemolysis than patient with equivalent number of type 3 cells. So those patients may be relatively less symptomatic than patients who are having the same percentage of RBCs in type 3 area or type 3 type. Okay, so that is the reason why you need to report type 2 and type 3 separately also. Okay, so here you find type 3 50% and type 2 is 0.2% and type 1 is 94.8%. Okay, this is before treatment. This is because most of the type 3 RBCs, though it is appearing relatively smaller, it is false because before treatment the patient will have hemolysis and all the type 3 cells will tend to die or will die in 7 days or 5 days. So you will not find a big population here. But once you start treatment with eculizumab, it inhibits complement mediated lysis. Now these RBCs are not destroyed by the complement and you see the relative proportion now. It is 91.2%. So such a big PNH clone is there. That is why we tell you it's always you need to check the PNH clone first on WBCs and not on RBCs. Because hemolysis can give you a false negative or give a false decreased burden of PNH clone. Understood? So that is the reason why we need to check on WBCs and then on RBCs and not vice versa. Okay, any doubts with PNH analysis on RBCs?
Sir, in that graph of forward scatter versus side scatter logarithmic scale, where do we see WBC, sir?
You will not see WBCs because the threshold is set in such a way. So you find RBCs here. You may find some WBCs. Okay, you will not. This plot will not cover WBCs. WBC will be out of scale. You will be setting the threshold of forward scatter and side scatter in such a way for an RBC assay, so that you find only the RBCs. So WBC will be out of the scale, so that it will not get confused.
Okay, sir.
Any other doubts? If you have flow cytometry in your lab or if you're planning to buy a flow cytometry in your lab, there are certain assays which you need to do. First is TNP, TBNK analysis, count, HLA-B27, PNH, CD34. Okay, so that is the reason why I've included all these things in the basic flow cytometry course. Leukemia, lymphoma, myeloma, MRD, these things are all a bit complicated. Okay, and then but it needs more expertise. But once you buy a flow cytometer, these assays have to be set in your lab. Okay. Right. Now let's go for analysis of leukocytes for PNH clones. In fact, this part should have come front and the RBC should have come later. But since RBC analysis for PNH clones is more technically demanding and little complicated, I want to cover that first. And then I'm covering the leukocytes. But the dictum is, first you need to check the PNH clones on leukocytes. If it is negative, you can sign it out as negative. If it is positive, then you can check it on RBC. So the leukocytes are the very important one or the gold standard one for checking the PNH clones, not the RBCs. Only why? Because hemolysis can false negatively give you, can result in false negativity, or it can active hemolysis can also give you a false decreased percentage of PNH clone when you are detecting in RBCs. So always you do PNH clone detection in WBCs, later on RBCs. Okay. Now, so as we saw with RBCs, here you need some gating markers. So someone can tell me what are the gating markers we can use for neutrophils and monocytes? Some wild guesses, no problem.
11
For monocytes, CD64.
Monocytes we can use CD64. Very good, excellent. Anything else for any other markers or for neutrophils?
CD13.
Very good, CD13. I agree with you, but I'll tell you why we won't use CD13. Any other thing? Any other? I appreciate that. Up 33, but yeah, good 33. Okay, now I'll tell you why we will not use CD13. See, we need a marker which is expressed in mature or only in the neutrophils. See, if you take CD13, CD13 is a myeloid marker which will be expressed even in the blast stage, even in promyelocytes. Okay, so that marker is not a pure neutrophil marker, which is expressed only in neutrophils.
So CD11C for neutrophils.
You can use it for CD11C for neutrophils, but it will not, even monocytes are positive for 11C. So how do you segregate neutrophils and monocytes? Of course, with scatter you can do that. And 11C is positive in some lymphocytes also. So we may not, 11C is not a very good idea. Okay. The best marker to get out the neutrophils is CD15. Okay. CD15 expressed well in neutrophils. The separation between the signal and the noise is very good. So you can use CD15. Okay. So that is one thing. For monocytes, as someone said, CD64 is a very good choice. And you can also use CD33 because monocytes will show bright CD33. Okay. So for gating the neutrophils and monocytes, first what I do, I use site scatter CD45. So I'll gate the neutrophil population by virtue of its high site scatter and CD45 moderate. Then I'll take out monocytes from the monocyte region in site scatter CD45. Then I'll apply the neutrophils which are gated with CD45 site scatter with site scatter CD15. Now I can purify and take only the neutrophils and I can take it for further analysis. Similarly, the population which I have taken from the monocyte region in site scatter CD45, I apply it on 64 or 33. The population which is expressing bright 64 or bright CD33, I can take it up for further analysis. And that's how I do gating for neutrophils and monocytes. For RBCs, we used glycophorin 235A. For monocytes and neutrophils, we can use in this way. Okay. For high sensitivity analysis of leukocytes, gating on light scatter alone, that is forward scatter and side scatter, may not be good. Okay. Or even light scatter plus CD45 is not acceptable because spurious events might be contaminated in the gate, leading to false conclusion that a PNH clone is present. So what they mean to say is like, if you are taking the neutrophils only by virtue of forward scatter, side scatter, or by side scatter 45, you may include some non-neutrophil population into that gate. And when you check it for FLAIR and 24, you may find some population which is negative for 24. And you may think that that is a PNH clone of neutrophil. The problem is that is not even a neutrophil. Okay, so that is the reason why they don't advise you to take the neutrophils and monocytes only from scatter plots or from scatter CD45, site scatter CD45. So you should include a gating marker into your assay or into your panel. So for the most accurate gating lineage markers such as CD15 for neutrophils and CD64 or bright CD33 for monocytes are generally employed. Combining site scatter properties with CD45 and these markers, now you can purify the neutrophils and monocytes in the best possible way. And you can take it for analysis for PNH markers. Okay. So this is the antibody panel combination for PNH analysis on WBCs, and they have given it for different machines with different colors. Okay. So if you have a 3-color instrument which is no longer used, okay, you need to design a tube separately for neutrophils or granulocytes and a separate tube for monocytes. If you are using a 4-color instrument, what you can do, you can design a tube for granulocytes separately, which includes FLAIR, CD24, which these two will be PNH markers, and one gating marker which is CD15 and CD45. Okay, so with site scatter CD45, you can get the neutrophils and then purify it with CD15 and then check for neutrophils for the expression of FLAIR and 24. Similarly, you can use a 4-color, another tube for only for monocytes, where you use CD45, gate the monocytic population from site scatter CD45, select only the population which is exhibiting bright CD33 or CD64, you can also use 64 here, and then check the purified monocytes for the expression of FLAIR and 14. So if it's 4-color, you can put 2 tubes, one for grants, one for mono. Or if you want to finish the assay in one tube where you include all the markers for granulocytes and monocytes, then you need to compromise on CD45. You need to have only CD33. With CD33, bright population you should take it for monocytes, and population which is positive but not bright has to be taken for granulocytes. And then check the respective population for 14 and flare for monocytes and 24 and flare for neutrophils, which is this one I will not advise because 45 is not there. If it's a 5 color, this is more optimal where you can beautifully do the assay for granulocytes and monocytes with one single tube, where you can include flare 24 for neutrophils, flare 14 for monocytes. You can use CD15 for neutrophil gating, and you can use CD45 for site scatter CD45 for locating the neutrophil population and monocyte population. Instead of 15, if you use 33, it is fair enough. Okay, but the best bin is 6 color. 6 color, no problem at all. You find 15, so you have 45, site scatter 45 to get the neutrophil and monocyte population. Then the neutrophil can be purified for 50, and such purified neutrophils can be studied for flare and 24. Similarly, you get the monocyte population with site scatter 45. Once you get the monocytes, purify the monocytes with CD33. So only you take the bright CD33 expressing population and check the purified monocyte population for expression of CD14 and flare, and that will give you the thing. So the best is 6 color, or with 6 color 1 tube panel, or a 5 color 1 tube, but you need to compromise on one of the markers here. 4 colors if you are doing, you need to put 2 tubes, one for monocyte and one for neutrophils. Okay, now this is how we do the analysis for neutrophils in a 4 color. So 4 color has only 4 markers: CD45, CD15, flare and 24, right? So first you eliminate the debris and take only the viable cells. Then you get that population in site scatter CD45. You take only CD45 positive population. After taking only CD45 positive population, you put it in site scatter CD15. Now you see CD15, how beautiful it is. The neutrophils or granulocytes positive for CD15 with high site scatter is taken. Now this is pure neutrophil population. Once you get it, once you have selected or draw a region around this population, you get it for 24 and flare, right? So you find, okay, this is lymphocyte control. This is lymphocyte control. Okay, so you get this population for 24 and flare. You cite beautifully. This is normal population or normal neutrophils, whereas you find a big PNH clone of neutrophils which is negative for flare and 24, which is around 91%. Okay. Similarly, another tube for monocytes. So similarly, you take the viable cells, select only CD45 positive events, and now you get this population in site scatter CD64. Select only the population which is expressing CD64, and now get this population across 14 and flare. Okay, so you find similarly a small population of normal monocytes and a huge clone of PNH monocytes. Okay, now let me tell you certain things. The PNH clone in monocytes will be always larger compared to neutrophils. Okay, the PNH clone in monocytes, see, for example, here it is the other way around, but regularly it will be the monocyte PNH clone will be greater when compared to the neutrophils. And whenever I'm saying 2 PNH markers, I'm telling you use flare and 24 for neutrophils or flare and 14. So the idea, the point here is you should detect PNH clone by 2 PNH markers. You should not detect PNH clone with only 1 PNH marker. For example, you don't have flare. You can't use only CD14 to detect the PNH clone. Getting my point? You should use 2 PNH markers to detect the PNH clone. Let it be neutrophils, let it be monocytes. That is the reason why I'm telling flare will solve the purpose for both monocytes and neutrophils. 14 will be used only for monocytes, 24 only for neutrophils. As I said, you 157 you can include, even that will help you for both neutrophils and monocytes. So the point here is you should show PNH clone with respect to 2 PNH marker negativity. Or 2 PNH markers. You should not use 1 PNH marker to detect the PNH clone. That is wrong. that is away from the guidelines. Okay, now this is a 5-color tube where you can do neutrophil and monocyte in one particular tube itself. Okay, so they have gated the CD45 positive events, then they have all the CD45 positive events is put across CD15. So only CD15 positive population is checked for 157 and flare. See, here they have not used specific neutrophil and monocyte marker, which is 24 and 14. Instead, they have used 157 and flare. So now you find the PNH clone, which is negative for 157 and flare, and normal population here. Similarly, monocytes, you get the bright, CD64 population, put it across 157 and flare. You find the PNH monocytes and normal monocytes. So here 5 markers they have used in one tube. Flare, 157, 64, 15 and 45. So flare and 157 will help as a GPA markers, 64 for monocyte gating, 15 for neutrophil gating and 45 as a backbone. Okay, now this is a 6-color assay where similarly they get the CD45 positive events, put it across CD15 and 33. So CD15 positive population are all granulocytes and CD33 positive population in a bright fashion is monocytes. After gating the neutrophils and monocytes, you put it across 14 and flare for monocytes, where you find the PNH monocytes, which are double negative for 14 and flare, and normal monocytes, which are positive for 14 and flare. Similarly, you get the neutrophils and put it across 24 and flare. You find normal neutrophils, which is positive for 24 and flare, and a big PNH clone of neutrophils, negative for flare and 24. Okay. So again, this is similar example. So this is control. And here you find the PNH disease. Okay, I'll show you live analysis with one of our cases. So this is control, where you don't find any PNH monos and grans, whereas this is a PNH case where you find the PNH monocytes and granulocytes. Okay, so report the PNH clone size in both the lineages for the white blood cells. That is PNH clone in neutrophils, how much you got, PNH clone in monocytes, how much you got, you have to report both of them. PNH monocyte clone is often larger than the granulocyte clone, and thus reporting only on PNH granulocyte may underestimate the PNH clone size. So you need to report both PNH monocytes and neutrophils. Granulocytes and monocytes may show presence of type 2 population. We studied type 2 population in RBC, right? Similarly, type 2 populations can be seen in neutrophils and monocytes. But in contrast to RBCs, the clinical significance of type 2 population in leukocytes has not been well established. So you need not give type 1, type 2, type 3 in leukocytes as you give for RBCs. Here you just need to say PNH clone is a percentage of PNH clone detected. In contrast to RBCs, where you need to tell how much is type 3 percentage, how much is type 2 percentage. Here you need not do that. Okay. So here is an granulocyte where it is PNH complete absence of flare and 24 in neutrophils. There is normal population which is showing 24 and flare, and a subset of population which is in between these two. So this is type 2, but still the clinical significance of this population is not known. So you can just give the disease combining these two. So PNH clone detected in neutrophils around 60.9%. You can just give like that. You need not give type 3 so and so, type 2 so and so as you give for RBCs. Okay. So establishing the sensitivity of the assay, as I said, if you want to achieve only 1%, you can just acquire 10,000 events or 25,000 events. But if you want to go to 0.01%, you need to acquire at least 5 lakh events. Okay. So this is called LLOD, LOQ experiment. I'm not going very deep into that. That may be a bit confusing. But if you want to do high sensitivity PNH, you need to acquire larger number of events. Somewhere around 1 million events has to be acquired to achieve a sensitivity of 0.005. Okay. So again, this is like dilution experiments. Okay. Now someone asked the doubt. So here comes the answer. How to interpret the terminology of reporting PNH clones. When you find the PNH population, okay, greater than 1%, you need to call them as PNH clone detected. Okay, when you're finding a PNH population in neutrophil or monocyte, whichever is greater. For example, in granulocytes, you're finding 0.8% and monocytes, you're finding 1.1%. Still, it will satisfy this. So PNH population greater than 1%, then you need to use the term PNH clone, the word PNH clone detected. When the PNH population is between 0.1% to 0.1% to 1%, then you need to use the term like minor population of PNH cells or minor PNH clone detected. Okay. If you are finding PNH population less than 0.1%, then you can put the word rare cells with PNH deficiency or rare cells with PNH phenotype. You will not use the word clone. You will use rare cells with PNH phenotype found or detected. So that is how you need to put your report terminology. Okay. So final impression in the report should be PNH clone present or absent. Okay. So you should not use words like potentially misleading, like a report stating that CD59 test is negative, which may imply that the target population is negative for marker CD59. So you should not use these ambiguous terms like negative, positive. You should report in crisp and clear, like whether the PNH clone is detected or not, present or absent. Okay, present or absent. Even you should not use the word like detected, undetected. So you may confuse that whether the clone is detected or if the marker is detected. Okay, so you should not use such terminologies. Okay, so always report the final impression as PNH clone present or absent. Don't use positive, negative or detected, undetected, which is not advisable. Okay, so this is how a model report should be given. So when the patient, you did not find any PNH clone, you need to report like no phenotypic evidence of paroxysmal nocturnal hemoglobinuria. Okay, no phenotypic evidence. Okay, so that means negative for PNH clone. If you find it, you put it as PNH clone identified in both WBCs and RBCs. And you see, you need to give the percentage. For RBCs, you need to give separate type 2 and type 3 and total. For WBCs, you need to separately give for monocytes and granulocytes. But this percentage is not mandatory. It's not needed. Okay. Similarly, when the clone is smaller than 1%, PNH clone identified in WBC, minor population of PNH clone identified in RBCs. Okay, so these are some of the reports how you have to give. Okay, so with that, we'll just finish and go to live analysis. Any doubts? Any doubts before we go into the analysis? I'll just stop sharing. Any doubts? Shall we go for analysis?
Sir,
Yeah.
you had told that for neutrophils and monocytes, we should use 2 markers always for PNH diagnosis.
Correct.
And RBC, 1 can be used.
Yes, but I also said you that you always need to start with PNH analysis on WBCs. Once you have found a clone, then you can check it on RBCs.
Okay. So it's basically to increase the sensitivity of the assay.
Yes, because once you find a PNH clone with WBCs, then you can check it on RBCs, because you can able to tell the clinician what type of RBCs is there, how prone the patient is for hemolysis. Because if you find a bigger clone of type 3, then it means like the patient will be having more of hemolytic symptoms and he has to act immediately. If the patient is showing predominantly type 2 RBCs of the PNH clone detected is predominantly of type 2, then the hemolytic features may not be that heavy. So the patient may not be having very bad hemolysis. As simple as that.
Okay, sir.
Right. Any other doubts? So it's crystal clear. Perfect. So let's quickly see a control case and then we'll see a PNH case. Okay. So I'm just starting with analysis. So someone will be telling me how to start. So what's the first plot we need to draw?
Time plot.
Time plot. So time versus forward scatter. So there's a stable acquisition. Nothing wrong with acquisition. Fine. Second plot.
Forward scatter versus
Forward scatter and side scatter. Okay. So I have to check only get the viable cells. See, there's a lot of debris here, which is showing very low forward scatter and side scatter. So I need to eliminate that population. So now I'll take only the viable cells, eliminating all the dead cells. Okay. Now I'll get only that particular population for further analysis. Now I need to do, what is the third plot? Third plot. Third plot. Come on. After live gate, what is the third plot? Viable gate. Right? Come on, what is the third plot we need to draw? Universal plots.
Singlet plot.
Singlet plot. So you do with forward scatter peak versus forward scatter integral. Okay, now you need to select only the singlets, eliminating the doublets. Okay, so I have done that. Okay, now what is the next plot? Come on, next plot. After singlet, which plot we need to use? Side scatter CD45, right?
45.
So you are finding neutrophil, monocyte, lymphocyte, monocyte, and neutrophil population. Right? What is this population? Anyone?
Debris or
Debris will show so much of CD45. Debris you have eliminated now.
The basophil or
Perfect. That's basophil population. Okay, that's the basophil population. Now what I'm going to do? So my interest is neutrophils and monocytes. So first I'll take population which is showing high side scatter and CD45 moderate. So this is likely to be neutrophils, and then I'm going to select monocytes. So when you're drawing a gate, always be generous. Don't be very stringy to gate the monocytes. Be generous because you also have a gating marker. So there you can eliminate the non-monocyte population. Okay, so I'm including a bit of basophils also. Now let's start with neutrophils. Now this is even that is not neutrophil, but I have to refine it. Okay, so now this is a 1, 2, 3, 4, 5, 6, 7, 8 color. I mean 10 color. It's a 8 color single tube assay for PNH on WBCs. Okay, you see I am using 3 PNH markers for the detection of PNH clone. For monocytes, I'm using CD14, I'm using FLAIR and CD157. So I'm using 3 PNH markers for monocytes and similarly 3 markers for neutrophils, FLAIR, 157 and 24. Though advisable is 2, I'm using 3. But if it is enough, if you use 2, 3 is too much. I mean, like, good, but 2 is fine, enough. Now, neutrophils I need to purify, I'm using CD50. Okay, so you see, these are all some eosinophils. Somebody asked me why we are not using CD13. CD13, if you are using, even eosinophils will come into the neutrophil or granulocyte gate. And that is the reason why I am not using CD13. Similarly, CD16 also I can use, but CD16 is a GPA marker. So I cannot use it as a gating marker. So hence I am using CD15. Now beautifully the eosinophils are separated and only neutrophils are there. So I'll get the neutrophils. Now I label them as purified neutrophils. Okay, I'll give them green color. I'll remove this green. Okay, so neutrophils are purified. Now we'll finish off neutrophils first. Let me put the purified neutrophils and check for expression of FLAIR and CD24. You see, beautiful, all the populations are negative. Sorry, all the population are positive for flare and 24. I'm getting a small 5 events which is falling a negative, but are they really neutrophils? I need to check. No, the gate, that would be some eosinophils or something. So I'm just tightening my gate to eliminate that. Okay, now done. So no population is, there's no cluster of events which is showing negativity for flare and 24. So the entire neutrophil is positive for flare and 24. So should I just check with 15 quickly? 157. So even 157, I'm not finding any population which is 157 negative. Okay. So this is no PNH clone detected in, or no PNH clone detected in neutrophils. Now let's go with monocytes. So monocytes are here. So I'll just get the monocytes, give the name as monocyte. Okay. See, next, when I'm getting next case, you need to tell me. Okay, I will not tell the subsequent gates. So monocytes, now I need to purify the monocytes. So purification, I can do it by CD64 and CD33. Okay, now monocytes are CD33 bright and 64 moderate. So the purified monocytes will be like this. These are the purified monocytes. See, now you can see the some lymphocytes which I've included and the basophils I've included is all eliminated. Now I'll put them as purified monocytes. Okay. Now I need to check the expression of GPA markers and purified monocytes. So I'll draw a plot. So I'll drop 2 GPA link markers, flare and CD40, and then put the purified monocytes. Okay. So the entire population is positive for flare and 14. Okay. There are some events which is flare positive, but 14 is slightly dimmer, but once I get it, you can see that that population is slightly sneaking into the gate in the border. So that is some non-specific events. So you can ignore them. Okay. If it is falling right in the center, then that needs to be checked. But here it is some events. Though if I'm using 2 markers to purify, still there is some population, non-monocyte population coming into it. Okay. So that is the reason why you need to have purification markers or gating markers. Then I also check with flare and 157. There is some compensation. Okay, done. So flare and 157, now get the purified monocytes. See, beautiful. So this area is empty. So there is no PNH clone detected. Again, the area is empty, even in neutrophils it is empty. So what is the final diagnosis? PNH clone. What should you give?
Not detected.
Not detected or PNH control? Yeah, so not detected. Okay. Now the next case you need to tell me. I'll draw the plots. You need to tell me what should I do. Okay, first plot, what should I do?
Time
You just
plot.
time plot. So first time versus forward scatter, check for stable acquisition. See, this is not a stable acquisition. See, there is somewhere at the end of the acquisition, there is some problem. Okay, so that is the use of time plot. Now what you can do when you find that, you can draw a gate until there is a stable acquisition and eliminate area where there was no stable acquisition. Okay, see, you can, beyond this, there is some problem, right? So you draw a gate until there is a stable acquisition, and then you can use it for subsequent plots. Okay, for this I am not used because there was good stable acquisition, but here there is some problem, so I am just gating the populations where a stable acquisition has happened. Okay, so that is the use of time plot. So what is the second plot now?
Viability.
Viability. So forward scatter versus side scatter, and now I am not going to use ungated. I will be using only events that were acquired properly. So once I have done that, I want to select only the viable events. So I am eliminating all those events with low forward scatter, low side scatter, and low forward scatter events, and I am getting only the viable events. Once I have done this, give a dark color to that. So viable events is done. So I have viable events. Next plot.
Singlet.
Singlet. So forward scatter peak or forward scatter height versus forward scatter area or integral. So now these are all some doublets. I want to eliminate those doublets. So sorry. So I am selecting only the singlets. Okay. So done, singlets are done. So the next plot.
Side scatter versus CD45.
Perfect. Side scatter versus CD45. Done. So as we saw with the last case, we are having a high side scatter population, which is possibly neutrophils with some eosinophils, a small monocyte population, and a lymphocyte population. So I'll get only the neutrophil population. Okay, I'll just give the name neutrophil. Similarly, I'll draw a gate around monocytes with 45 side scatter. Again, I'll draw a more empirical or a broad gate, including some lymphocytes, which I can eliminate with the subsequent plots. Okay. Right. Now, what is the next thing which I need to do?
Side scatter
Purify
versus
the neutrophils.
CD50.
Purify the neutrophils. So I'll take the neutrophils. How do I purify them?
CD50.
Perfect. Side scatter versus CD50. Okay. Now you see this cluster is purified neutrophils, and these are some eosinophils and other populations. So I'll just gate only the purified neutrophil population. Okay. And label them as purified neutrophils. Perfect, done. So now we have purified the neutrophils. Now let's check for the expression of
Flare and 157.
perfect flare and 157 and flare at 24.
24.
Yeah, so I'll just put flare. See, you can also purify monocytes and then check, but we'll finish neutrophils first. Flare and CD24, and I'll put the purified neutrophils. Okay. Just a minute, there is some compensation with 124. So now you find that the neutrophil population is splitting into two in contrast to the last case, right? So you're finding a population which is negative for 24 and flare. Okay. So now we will also confirm that the same population with another PNH marker which is 157. Okay. Right. So you are finding a small PNH clone of neutrophils which is negative for flare, negative for CD24, and it's also negative for 157. How much is the percentage? So it is 7.2%. Okay, it is 7.2%. 7.2% of the neutrophils are PNH clone in or PNH clone. Okay, now so this is there. So we have found a PNH clone in neutrophils quantified at 7.2%. Okay, now we'll go with monocytes. Now again we need to purify the monocytes. So I'll drop monocytes. Now how do I purify the monocytes?
64 versus 33.
Yeah, you just use 64 versus CD33, or if only CD33 is available or 64 available also, it's fine. So now the monocytes have clearly separated out from lymphocytes and other population. Now get the monocyte population. Okay, now I label this as purified. Right. Now the next plots for detecting the monocyte. So flare and CD14. Similarly, you use flare with CD157. Now drop the purified monocytes. Correct? So sorry. It's the first thing I need to change. Okay. Now drop the monocytes. Once you drop the monocytes, you find there are two populations. One is normal population expressing 14 and flare, and there is a clone of monocytes which are negative for flare and 14. So check with the percentage. Yeah, MFI I need to tell. Yeah. Okay. So 12% is this particular population. So you can also check in this gate also. These are all statistics, just getting the population. Okay. So 12% of monocytes are PNH clone and 86% are normal monocytes. Okay. Now similarly, I'll just check with purified monocytes and with 157 and flare. So am I getting the similar percentage here also? So 11.9%. Here it is 12.08%. Here it's 11.9%, almost 12%. So 12% of monocytes are PNH clone. Okay, so PNH clone is detected in monocytes quantified at 12%. Now, how do you report this case? What are the terms you use? You will say PNH clone detected. Okay, and what is the percentage you will put? You should use the maximum percentage. Okay, PNH clone detected at
12%.
12%. And you also need to give separately PNH clone detected in neutrophils and PNH clone detected in monocytes. And then the final report does. Okay, and you should give an impression like evidence of PNH detected in neutrophils and monocytes. Evidence of PNH clone detected in neutrophils and monocytes. And then you need to give the percentages. That fulfills your report. Okay? Any doubts? Any any doubts?
Sir, in positive cases, we'll not check it on RBCs also.
You need to check it on. But I'm not doing it. It's a better practice. It's a good practice for a positive case to check it on RBCs, as I said. But I am not doing it currently. I need to implement, and it's a better practice. If it's negative, no need to check RBCs. But if it's a positive clone is there in the leukocytes, that is monocytes and neutrophils, you need to check it on RBCs. You need to do that. I am not doing it, but it's better you do it. Any other doubts? So I'll show you what is MFI. So since there was a doubt on that, see, MFI is median fluorescent intensity. Now, for example, I need to check the median fluorescent intensity of neutrophils for CD15s. What I do a histogram, get the population of needed. So, for example, neutrophils, purified neutrophils, and then the marker which I want to see, which is 15S. Okay. Now, if I want, so I can say that the neutrophils are expressing CD15S in a moderate fashion, in a moderate intensity. But to give it in a numbers, to be very precisely calling it, okay, you use MFI. So, if you're using a software for analysis, it is an automated one. So, in this software, if I right click and if I click this percentage symbol, it gives me all the statistics. So, if I want to check the median fluorescent intensity, I just need to click the median. So, the X median is around, here it is a number. Okay. So, similarly, you can do it. Okay. For example, just a minute. So, here it is a composite file, so the number appears a bit larger, bit bigger in number, but so that's how you check for MFI. If for just I'll drag one more file for you. So just for MFI, I'll just show you. So for example, now I just want to check the MFI of the granulocytes for flare. Just right click it, select the percentage and click on the median. So it is 31.64. If I want to check the MFI only for the positive population, it is 32.626. So this is how you check the median fluorescent intensity. So similarly for median, you can check the even mean fluorescent intensity where you select either the arithmetic mean or geometric mean. So this is mean fluorescent intensity. This is median fluorescent intensity. This is mean. Normally we use median. Okay. Similarly, other parameters include CV, half peak CV, okay, mode, standard deviation, all these things are there. Okay. So, Dr. Nima, you understood what is MFI now?
Yes, sir.
Clear? Any other doubts you have?
No, sir.
So, other participants, any doubts?